Now that we have looked at the different options for your primary antibody specificity, we can move on to validating selectivity. The selectivity of an antibody within the context of a Western blot means that the antibody finds its target in the presence of many other proteins in the same area on the membrane. An antibody is considered sufficiently sensitive if it can detect its antigen with high affinity at low working concentrations. An antibody that has low affinity for its antigen will bind only at higher concentrations. This means you would have to use a lot more antibody to achieve reliable results. Binding of the antibody to its target is notably influenced by assay parameters. Seemingly small changes in a Western blot workflow, such as membrane choice, blocking buffer, incubation temperature, and primary and secondary antibody dilutions, can create unpredictable and perplexing outcomes. This is why setting a protocol that details this information for each antibody is critical for reproducibility in Western blots. The dilution or dilution range provided by the vendor for a Western blot should be considered a starting point for optimization by the user in their experimental conditions. The goal of optimization is to identify the highest signal-to-noise ratio with the lowest background. The benefits of optimization are the most efficient use of antibody and validation of antibody selectivity. To identify the optimal antibody concentration that will give you the highest specific staining and the lowest background, you need to do a twofold dilution series of the antibody. Have a minimum of 5 dilutions with the vendor's suggested concentration in the middle. For example, if the datasheet suggests a 1 to 1,000 dilution of the stock antibody, your range should be 1 to 250, 1 to 500, 1 to 1,000, 1 to 2,000, and 1 to 4,000. Purified proteins on their own or spiked into samples and samples overexpressing target protein have a bias toward your target. Antibody binding to your target in this artificial setting is not acceptable proof of validation. Validation should be performed using samples with endogenous levels of protein expression. Secondary antibodies need to be optimized as well. High concentrations of secondary antibody can increase off-target interactions, resulting in high background. Low concentrations of secondary antibody can exhibit faint bands that are below the level of background. The optimal concentration for the secondary antibody relies on the type of conjugate and the detection method.